Fermentation is a metabolic pathway that incorporates microorganisms to convert carbohydrates, like sugars, into other compounds including acids, gases, and alcohols. This results in an anaerobic energy release. There are three main types of fermentation: alcoholic fermentation, lactic acid fermentation, and finally acetic acid fermentation. Acetic acid fermentation involves the process of converting ethanol into acetic acid. This conversion is completed due to the ethanol undergoing a reaction with acetic acid bacteria, usually Acetobacter or Gluconacetobacter. This reaction is an oxidation reaction with end products of acetic acid and water. Acetic acid fermentation is usually completed under conditions of a temperature of around 25 to 30 degrees Celsius and in slight acidic conditions with a pH of around 4 to 6.
Alcohol fermentation uses fructose, glucose, or sucrose from sources such as grains or fruits, which is inoculated with yeast. This first step is also glycolysis, which produces two pyruvate molecules. These pyruvate molecules then undergo decarboxylation, where they are converted into acetaldehyde and carbon dioxide as the byproduct. Lactic acid fermentation uses substrates such as glucose and lactose inoculated with bacteria such as lactobacillus as the microorganism. Glycolysis is the first step in which glucose molecules are converted into two pyruvate molecules. These pyruvate molecules are then converted into the desired product of lactic acid by an enzyme called lactate dehydrogenase with byproducts of a phosphate group and two ATP molecules.
Fermentation can be used to create dairy products or even bread. The production of alcoholic biofuels is also another use for fermentation to help fuel cars, as well as curing tea, tanning of leather, and preparation of antibiotics. These are a few more uses for fermentation. By far one of the most important uses for fermentation would be glycolysis. The regeneration of NAD+ is essential because it ensures that glycolysis can continue. If NAD+ were not regenerated, glycolysis would be halted due to all the available NAD+ would be reduced into NADH, and there would be no NAD+ to accept new electrons. By regenerating NAD+, cells can keep glycolysis continuing, ensuring a continued supply of ATP even when oxygen is scarce in anaerobic conditions. Overall, we've discussed what is fermentation and different types of fermentation. We can conclude that fermentation is not only used for cellular energy production, but can also be used in economic aspects as well as cultural.